As we move from collecting water samples in the field to sample analysis in the lab, it's important to talk about how to ensure those samples provide quality data. After the samples are collected in the field, you have to transport them to the lab. These transport details are important to document and report, for example, in our journal articles, because these details are indicators of the quality of our data. We generally transport water samples on ice because the little microbes that are present in the soil can be present in the water samples too. Keeping the samples cool by transporting on ice keeps these microbes slow enough so they don't change the forms of nitrogen, for example, in the water. If the water chemistry or the forms of nitrogen in the water change between when you've collected the sample and when you analyze the sample, I think you can see that your results wouldn't be reflective of the actual water that you were trying to sample. Moving on to sample preservation and storage, just like sample transport, these two components are important details to report because they're indicators of the quality of your data and the quality of your process. The standard methods for the examination of water and wastewater is the standard, literally, for water sample preservation and storage procedures. In our lab, we normally use pH control or acidification, refrigeration, or freezing the sample. You should note that freezing a sample prior to analysis is not the preferred method. That's where you should consider what is your timeline for the preservation process that you're doing. Are you trying to keep a sample for just a couple days before it gets analyzed? Or has the sample already been analyzed and you're storing the sample more long-term in case it needs to be reanalyzed? My lab's policy is to keep all samples until those data have successfully undergone peer review and the journal article is published. We do that because reviewers sometimes do suggest you analyze samples for additional constituents. Now, that's not always possible, but if you have stored the sample appropriately, you'll at least have the option of doing that. You see this quote from the standard methods: "No single method of preservation is entirely satisfactory. The best preservation method is, in fact, to not preserve the sample at all by analyzing the samples immediately. When you're able to do that, you remove all uncertainty or error that is inherent in having to preserve a water sample." Here's a screen clip that is obviously too small to read. This table from the Standards Method book lists the recommended preservation methods for a number of analytes. If you are doing water sample analysis, I highly recommend you check this table out. Let's zoom in on nitrate, which in our lab we analyze as nitrate plus nitrite, and you see the recommended preservation method of acidification to a pH of less than 2, or keeping the sample below 6 degrees Celsius. Ideally, you are able to analyze the sample within 48 hours of collection. That almost never happens in our lab, but we do aim for the regulatory maximum storage of analyzing the sample within 28 days. Note that regulatory column is really important for point sources like wastewater treatment plants who have strict reporting terms on their legally regulated discharge permits. Another thing I want to note here is that the acidification method recommends use of sulfuric acid, but remember, back to the previous slide, choose the preservative with due regard to the determinations to be made. We discovered that we can't use sulfuric acid to preserve bioreactor water samples, for example, if we want to analyze the sample later for sulfate, which we sometimes do for bioreactors. So in those cases, we started doing long-term storage preservation with hydrochloric acid rather than sulfuric acid. Moving on, you see for dissolved phosphorus or phosphate, the standard method says you need to filter the sample immediately. That generally means you need to filter the sample within 15 minutes of sample collection. We, along with many other drainage research labs, do not actually do that because it's often not possible to do this in field studies where your field sites are far away, unless you're actually manually grab sampling on site. And you can use an in-field filter syringe, which some labs do. In this case, though, the important thing is that we fully report the method we do use so that readers can assess for themselves the level of uncertainty our dissolved phosphorus data present. You always report your method. Another thing about sampling water for phosphorus is that the container matters. Dissolved phosphorus has a high affinity to adsorb to container walls, and so this reference recommends acid washing polyethylene bottles. And coming back to the standard methods, you see the standard method is to sample phosphate into glass bottles, G, that have been rinsed with nitric acid. Side note, you also see minimum sample sizes listed here, which we discussed in a previous lesson. The standard methods book is really just such a gem if you're doing this kind of work. Once we're back in the lab, we filter our water samples as quickly as we can and always within 24 hours. We filter because we're interested in the dissolved form of nutrients for most of our tile drainage studies. Samples collected for analysis of total phosphorus or total nitrogen do not get filtered. So if you want to analyze for both total and dissolved phosphorus, for example, you would want to collect enough sample to split the sample, or you would need to collect 2 separate samples. If you're interested in total suspended sediment, there are additional steps like weighing, drying, and re-weighing the filter paper to know how much sediment has filtered out. One last note about filtration. This process is required for determination of dissolved nutrients in water, but the process itself can change the sample. How frustrating is that? For example, the mechanical effects of filtering under a vacuum can rupture cells, which can release dissolved nutrients into water. So thinking back to the data uncertainty lesson in the last module, the process of filtering, which is absolutely necessary if you want to analyze for dissolved nutrients, does introduce uncertainty. And one last note about filtering, we have always used a 45 micron filter to filter water samples, and this is pretty standard for drainage research labs. Now lastly, the most glamorous part of working in a water lab is the fact that this type of work requires quite a bit of bottle washing. Every water lab will have its own standard operating procedure, SOP, for bottle washing, and I'm sharing ours with you on the screen. With that, see you next time.